And clear?
Yes.
Wonderful. And Sergio, just to double check, so it's roughly 50 minutes, right? I
Yeah.
mean, okay. You tell me if I'm going over time, okay? I
Sure.
will, I haven't rehearsed much. Okay, everybody. So, well, I must say that thank you to begin with. Thank you for giving me this opportunity. Thank you, Sergio, for inviting me to give this guest lecture. And thank you guys for being here with me today to learn a little bit about genome evolution. So I just want to preface by saying that this lecture is based on a course I've taught 2 years ago. In here, we have Bio 4563G, called Genome Evolution. I taught this course in the winter of 2023. So basically for today, I tried to create a crash course, in one lecture only, all what I've taught in one term. So I want to acknowledge that I will be not only speaking a lot, but I'll be covering a lot. Mostly, I want to say at the superficial level, but I'm hoping that I will be eloquent enough to teach you anything about genome evolution today. And I just want to very briefly talk a little bit about Western. So here is a aerial picture, one little place of a piece of our campus. It's a beautiful campus. Anyone who would be interested in coming here one day, let me know. You're more than welcome. I'm actually not that far from you guys. As you can see, I'm in between Toronto and Windsor, close to Detroit as well. And before I actually start the lecture, I want to acknowledge, the caretakers of the land I'm speaking from. So I acknowledge the Anishinaabe, Haudenosaunee, Lenape, and Adirondack peoples whose traditional lands I present from today. And if you are interested in knowing a little bit about the indigenous peoples of your region where you are located, I invite you to take a look at this website, native/land.ca. Even though it is a Canadian website, there is information about indigenous peoples across the globe. With that, let me see. Oh, I knew. Okay. Well, again, I'm Matthias. I'm a PhD candidate in biology. I just started my 4th year, and we do a little bit about genome evolution here in our lab. This beside me here is David. He's my supervisor, a professor in biology here, and we are, I want to say, each other's partners in crime. So we have a lot of fun here in the lab studying genomes. And if you are interested in knowing what we do, here's our website, arrogantgenome.com. You can see our publications. You can see David's work with science communication to large audiences. And you can see a little bit about as well other team members of our lab. So I invite you to take a look at our website. And last but not least, I want to just plug in a evolution seminar series that I'm a host of. It is on YouTube, and pretty much what I do in this seminar series, I invite speakers just like Sergio did with me. I invite speakers in different moments in their careers from different places, and I record those talks and then I post them online on YouTube and also on our website, which is evobioseries.com. So if you want to learn anything related to evolution, make sure to take a look at the website, subscribe to the channel if you will, and I hope you enjoy it with me. Now let's then get started. So for today's agenda, as I said, this lecture is based of an entire course that I've taught 2 years ago. So it was difficult for me to find I guess, a roadmap. And I want to say that during the talk, I will be talking about genome evolution, but in different levels. So I'm hoping to focus at the beginning mostly at the anatomy of genomes. So I'll be talking about genomes almost as if they were any morphological trait of any organism that you want to study. But you see that I will transition to genomic signatures of past evolutionary transitions. That transition may not be I may not acknowledge the transition as I speak, but you see that I moved to that. And then in the end, I really finished talking about genome evolution per se, almost as an overarching theme in which I sprinkle some pop gen concepts in there to end up with genome evolution. But I have those 3 layers here all connected because at any given time in this talk, I might be moving between layers at different moments. And I'm hoping that this will be, again, intelligible and eloquent enough for me to convey a compelling message today. So to begin with, let me just start off with a brief history of genomics, because I think at this day and age, we often think about genome with massive sequencing technologies like Illumina sequencing, for instance. And precisely more recently, we have been talking a lot about long read technologies, which are, the most famous platforms really are PacBio and Oxford Nanopore. So currently we are at the age of T2T assemblies, which are telomere to telomere assemblies, but that hasn't always been the case. In fact, the field of genomics is fairly recent, is fairly young. And the first nucleotide to be sequenced was just in 1965. And it wasn't even, where's my, oh, here, is my cursor here. It wasn't even a DNA molecule. Actually, it was just a fragment of an RNA, because at that time, we already had the techniques for RNA sequencing, but not so much for DNA sequencing. And I want to emphasize that the first molecule to be sequenced wasn't even a nucleotide or a polynucleotide, I'm sorry, was actually a protein. So the first molecule to be sequenced was the insulin molecule, a few years before we had sequenced a partial tRNA from yeast. So we've come a long way, but I always think that for us to develop a real appreciation to any field, we should be putting our knowledge into a historical context. Now, again, starting from the basis, I just want to very briefly revisit the central dogma of molecular biology, and you understand why I'm revisiting this basic concept in a minute. So as we all know, the central dogma of molecular biology really states that unidirectional movement of information from DNA to RNA to protein. However, viral genomics, which I'm going to talk about in a second, in the early 70s, changed the first proposition of the central dogma, because when David Baltimore with other colleagues have identified reverse transcription in retroviruses, which are viruses that have genomes made of RNA only, this was when researchers at the time realized, oh, we need to amend the central dogma of molecular biology. So at that moment, which now is roughly 50 years ago, we realized that the central dogma had to accommodate for a bidirectional movement of information from RNA back to DNA and from RNA to RNA itself. Now here, DNA to DNA stands for replication, right? One thing I want to emphasize with this slide, though, is that we don't have the direct transition from DNA to protein and vice versa. We don't have reverse translation, so to speak. So that is one aspect of the central dogma that has remained true to this day. Now, speaking of viral genomes, so viruses are interesting and complicated because they aren't life forms, right? But what is interesting about them is that they have their own genomes, and their genomes are very diverse, not so much in size, but in the inherent molecular nature of these genomes. So viruses are the only entities that have genomes made of DNA or RNA, and their genomes are also can be double-stranded or single-stranded. We don't see this pattern with any other group, right? So there is, evolutionarily speaking, there is a lot to be said with this observation, which we can discuss about in our Q&A session. But here I'm just showing to you what's called the Baltimore classification of viruses, which was again in the early 70s when David Baltimore proposed a taxonomy of viruses based on how their genetic material, their genomes, are transcribed into messenger RNAs. Now, I will touch upon very briefly on three aspects of viral genomes that I find very interesting. The first one being what we call overlapping genes, which create what's called overprinting or overprinted proteins. So the overlapping of genes is not exclusive to viruses only. In fact, we see genes being overlapped in eukaryotes and organelles and prokaryotes as well. But what is interesting about viruses, and that happens with prokaryotes as well, is that the actual coding sequences, so the fragments of the gene that encode for a protein, they can overlap. And that's why we say those overlapping genes will then undergo what's called overprinting, which is the production of two different proteins from very similar coding sequences. So the same sequence is read in different reading frames, giving rise to two different proteins. We see overlapping in eukaryotes, but the overlapping regions of eukaryotic genes happens in fragments of the gene that does not give rise to proteins. So for instance, the untranslated regions of genes. Another interesting aspect of viral genomes is that they can be segmented and multipartite. And I'll explain to you in a second what I mean by that. But I want to say that even more interesting is that we can almost break down and establish a correlation between the material of that viral genome, let's say double-stranded DNA, single-stranded DNA, their configuration, whether it's that, the type, or the type of molecule, whether it's multipartite, segmented, or monopartite, and their more probable or more common host species. So there's a lot of evolutionary discussions about how those correlations explain the history of those viruses. But let me tell you a little bit about multipartite viruses and segmented viruses. Well, to begin with, to be multipartite, you need to be segmented. And basically, segmented viral genomes are those genomes that are made of different RNA molecules. We don't have, if I go back here one slide, you will not see DNA, viral genomes made of DNA that are segmented, okay? So those different molecules then, so let me just step back. The segmented genome are genomes made of different molecules, okay? Which could be, if you establish a comparison to eukaryotes, I'm talking about different chromosomes. But we don't use the word chromosome for viruses, we use the word segment. Now, segmented viruses can be multipartite, meaning that each and every segment actually is encapsulated in a different capsid. And they will be transfected, they will be transmitted separately from one another. Now, I do understand this figure here is a little convoluted, but the idea here is just showing us three different theoretical models of how those multipartite viruses, in fact, proliferate in the host and are also reassembled. Because evolutionary speaking, it is very challenging to understand how one species has its genome separating different instances of this species and how they come together in the end in a host species. So again, I'm just briefly touching upon this, but just to give you an idea of the genomic diversity. Now, here I have, again, an example of a segmented RNA virus. This is influenza virus. And as we can see, each and every segment represents a RNA molecule, which is one piece of the genome, which is equivalent to chromosome, if you will. And it's in this particular virus strain, those segments are organized in this 7+1 arrangement. But again, I'm just giving you, broad strokes, broad brushes of this idea. Moving forward to prokaryotes now. So now we're going to talk about cellular life forms. You will see similar graphs like this throughout my talk, because to understand genome evolution and genome diversity, I think it's really important for us to understand genome size variation. And you see later on when I talk about eukaryotes, how genome size variation has been puzzling scientists for decades now. Here I'm showing you a direct correlation between genome size and number of proteins, both in bacteria and archaea. And what I want to show you here with this graph is that we have almost a perfect correlation between genome size and number of proteins, meaning that those genomes, as they grow, they are growing because they are accumulating more protein coding genes. You will see later in my talk that that's not the case for eukaryotes, okay? So there is something to be said about the population genetics, dynamics of those species, of those communities, and the evolutionary pressures that explain this pattern. Now, again, very briefly, I just want to point out, that, for instance, if you plot here genome size and the frequency, so meaning how many species have genome sizes of these sizes that are shown here, we can see that archaeal species tend to have slightly smaller genomes, and bacteria then will have a a broader spread with some instances of relatively large genomes for prokaryotes. Again, we see that there is a very similar distribution of gene length, both in bacteria and archaea, and there's a relatively similar distribution as well of the ratio of intergenic regions in those genomes. What is interesting, though, is that archaea shows a faster accumulation of intergenic regions as their genomes grow, which I actually, when I was reading it, I was looking at this graph again, I realized that this is very interesting because evolutionary speaking, archaea is closer to us. You see actually that, I mean, phylogenetically saying we are archaea, and I'll explain to you in a second later. Sergio might have touched upon that eventually in his course, but this pattern here we see in archaea is closer to what you see with eukaryotes. I just want to remind us that the quintessential signature of prokaryotic genomes is the operon. And here I'm just showing you the LAC-Z operon, which is the most famous operon. It was really the workhorse of genetics. This operon was the subject of study for us to understand gene regulation decades ago at the beginning of the field of genetics. But we have more than operons in prokaryotic genomes. In fact, it depends on the level of resolution you're studying that genome. So operons will be here in this level of resolution, right? So operons, they sit a little above genes because I should have defined what operon is. So operon is when you have multiple genes sitting side by side. Usually they are on the same direction, and usually they encode for proteins that are under the same biochemical pathway. That is not always the case, but that really is the trend in operons, right? And all these genes within one operon are under one common regulatory element here. So that's why operons, the resolution of operons sit above genes. But again, you can study domains in prokaryotic genomes, repeats, motifs, islands, you name it, replicors, you name it. What is very interesting about prokaryotic genomes, which I really like to compare with eukaryotic genomes, is that it is difficult to determine, or let me put it this way, it is difficult to predict the gene complement of prokaryotes because of rampant horizontal gene transfer, which I'll talk about in a second, but it is very easy, or we have strong confidence in predicting the actual physical structure of prokaryotic genomes because they are pretty much always like this. Now, of course, we are studying biology. There's always exception to the rules. So we do have, for instance, bacteria species with linear genomes, but usually we have prokaryotic genomes being circular mapping. They have just one origin of replication. They have strand-specific asymmetries. So most of their genes are sitting on one strand only, which happens to be the leading strand, which we can talk about it in the Q&A session, why that is the case. And what is very interesting is that there is a replication-associated gene dosage because we have a lot of fundamental genes that are sitting close to the origin of replication, and these genes are being replicated more often, more rapidly than the ones that are sitting in the terminus of the replication, okay? So here are some patterns that are easy to detect and are easy to predict if I give you a fully sequenced prokaryotic genome. Now, I told you a few seconds ago that horizontal gene transfer is rampant in prokaryotes. And in fact, this actually makes phylogenetic studies of prokaryotes relatively challenging, particularly when we are concerned with species delimitation. I just want to, remind us of the multiple channels, the multiple ways in which bacteria and archaea can absorb foreign DNA, both environmental DNA, but also DNA from other species. So we have a gamut of alternatives that really makes prokaryotic species real sponges of foreign DNA. And this is not a new knowledge. In fact, from, I want to say early 2000s, perhaps even late 90s, we had already, and when I say we here, I mean mostly, the scientific community, not myself. So we had already realized that prokaryotic species were, again, absorbing a lot of foreign DNA. Here, whatever is in blue here shows the species-specific DNA. Whatever is in yellow shows transposable elements, and in light pink, if you will, reddish, is DNA acquired horizontally. So already in the 2000s, we had realized that there is a lot of foreign DNA in those species. And whatever is A here represents archaea. And over time, then, the more prokaryotic species that were sequenced, right? So their genomes were sequenced, the concept of pangenomes came about, right? So a pangenome is the collection of all genes in a particular species. So let's think about E. coli for a second. If we take different strains from different isolates, different geographic regions, and sequence the genome of each and every strain and compare their gene complements or gene repertoires, we're going to see that a group of genes is always present. This is called the core genome, right? It does not matter which strain I'm sequencing. I know I will find, let's say, 10 particular genes. But other than that, outside of the core genome, we have an accessory genome, which is the gene complement that varies according to the strain, right? So let's say, I'm hypothesizing, okay? So if we grab a strain from North America, I'm gonna have, 10 different genes compared to the one in South America or in Europe. So that is the accessory genome. And depending on how this pangenome, whether the pangenome grows or not, we say that we have an open pangenome or a closed pangenome. A closed pangenome is the case in which the more strains you sequence, the core genome does not change, and the size of the accessory genome does not change. Now, open pangenomes is the case in which the more strains you isolate and you sequence their genomes, you see that over time, the core genome shrinks because suddenly you realize that the common complement of genes in those different strains gets smaller and smaller. It is rarer and rarer, but then the accessory genomes gets more spread. So again, there's a lot. What I want to say is that for each and every aspect of genomes I'm talking here about, there could be a lecture on it, and there is a whole host of evolutionary concepts and ideas that can be derived from these facts. Now, moving to organelles. So here I'm saying organogenesis / eukaryogenesis because, eukaryotes have organelles, and in the birth of eukaryotes, we had the birth of organelles. Now, of course, I won't get into the nitty-gritty details of which one came necessarily first, but for this talk, let's assume that they happened together, right? And in the emergence of organelles, what happened? Let's first talk about mitochondria. So we had a archaeohost, right, which engulfed a free-living bacterium or several bacteria, and over time, that bacterial endosymbiont then relinquished its own control and became the mitochondria. So first we had the emergence of mitochondria, and eventually when we had the established, my cursor here, when we had the established eukaryote with mitochondria, billions of years ago, we had then the establishment of photosynthesis within eukaryotes. This was a second endosymbiotic event, and then which gave rise to most of our photosynthetic eukaryotes. Again, I won't get into the phylogenetic and biochemical details of these events, but what I want to tell you here is that organelles, meaning mitochondria and chloroplasts, once were free-living bacteria. What is interesting in evolutionary signature of this event, this transition from a free-living bacterium to a fully fledged organelle, is the unidirectional or asymmetrical gene transfer, which we call endosymbiotic gene transfer. So in both cases, mitochondria and chloroplast, the endosymbionts, they relinquished their control by I want to say again, I am, humanizing what has happened, right? So by giving their genes to the nuclear genome, to the host nuclear genome, um, most of these genes actually were lost eventually, but a lot of those genes then were transferred to the nucleus, and that really culminated in the transition of an endosymbiont to a fully fledged organelle. That happened for both organelles. And sometimes I'm looking here in my timer, okay? And in the current circumstance that we find ourselves is that we have a intricate communication between organelles and the nucleus, right? Which can be divided in retrograde signaling when the organelles are speaking to the nucleus, or anterograde signaling when the nucleus are talking to the organelles. Now, of course, this model here is showing both chloroplast and mitochondrion. This depends whether you're studying a photosynthetic organism or a non-photosynthetic organism. What I want to say here is that even though this communication is very intricate, right, and it does, we do have signatures of this communication also in the genomes of these compartments. I also want to say that, for instance, any signal that is coming from the nucleus hits all organelles at the same time. So it's one signal hits all. And the same thing here, well, from the organelles, if those signals are destined to the nucleus, there's just one nucleus to be speaking with. But these, we also have communication between organelles. So it's not, I don't want to say not precise, but it's not targeted, because there's no way to target just one organelle within the cell in this particular channel. Now, very, very briefly, organelle genome diversity. Well, when organelle genomes were first sequenced roughly 40 years ago, we thought that organelle genomes were going to be boring. They were all circular mapping. Mitochondrial genomes were small, around 16,000 base pairs. Chloroplast genomes were relatively larger, but also not very interesting. Well, fast forward now, I'm trying to show you here, to represent you here, the diversity of genome, not only size, right? So we have here, although it's not drawn to scale, I'm here trying to represent, very bloated organelle genomes and very tiny miniature organelle genomes. Whatever's in purple is mitochondrial genomes, whatever is in green is plastid. So I'm trying to represent here the diversity of size, but also of structure. So we know today that, in fact, most organelle genomes, even those that are circular mapping, in the cell, in the organelle, they will be in different configurations, most likely linear branched. But we have, linear monomeric with telomeres. I mean, telomeres, not precisely like the nuclear telomeres, but they serve the same purpose here. We have mini circles. We have also diversity in gene structure. We have diversity in nucleotide composition. We have diversity in intergenic region ratios. So there's a lot to be said about the diversity of organelle genomes, which I'm happy to talk about anytime with you, because this is what I study here in the lab. Moving forward, let's talk about eukaryotes now. And I do understand that I'm fast-paced here today. Again, I'm just reminding you, this is a crash course. So hopefully you will enjoy it in the end anyways. So before I talk about eukaryotic genomes, I just want to remind ourselves where eukaryotes are. And I'm assuming, you will learn more about phylogenetics later in this course, but I just want to convey the following message to you today. At the eukaryotic tree of life, we now talk about supergroups. So that's really the term we are using, right? So before, a supergroup can be interchangeably used with clades, but usually we talk about supergroups. And here we have some supergroups that you get to know later in your course. But what I want to emphasize more is that the latest tree of life really shows us that eukaryotes, they branch from within archaea, which again, archaea is a paraphyletic group, and phylogenetically saying we are within archaea. Now, I'll let Sergio explain to you the details of this to you later in another time, but I find this fascinating when, we're moving from a 3-domain type of tree to, if you will, a 2-domain type of tree with eukaryotes branching from within archaea. And that again, There's a lot to be said here about the evolution of these groups, of what were the evolutionary evidences for us to conclude those things, which we can discuss at another time. But let's talk a little bit about the genomes of eukaryotes itself, right? So here I'm representing to you genome size diversity in eukaryotes. We can see already straight off the gate that nuclear genomes are much, much larger than prokaryotic genomes and organelle genomes and viral genomes for sure. We're talking about gigabases, so billions of base pairs, billions of nucleotides. And what I find interesting is that it seems as though there is a physical limit to how big nuclear genomes can get. So around 150 gigabases, this really is where we have reached so far. But again, in biology, or I guess like anything in sciences, we can only speak about what has been discovered, right? You never know what's unknown out there. But we can see that most nuclear eukaryotic genomes are actually relatively small within this group. So, they are within 3 gigabases, 5 gigabases. So these are violin plots that show not only the distribution of nuclear genomes, but their frequency. So most, genomes are falling in those thicker bases, okay? Now here I'm showing you a comparison of the genome sizes of different eukaryotic groups, but also compared with prokaryotes. And we can see, and this is a log scale, right? So we can see that, genome sizes are changing in orders of magnitude. And we can see that eukaryotes, they span a gamut of genome sizes. And I just want to say, if you read this paper, you will see that the author, who is a Canadian genomicist, later on I'm going to talk about him, he put mammals here on purpose, right? He was like, look, I'm going to put mammals on top just as a joke because of our human-centric view of things. Now, although eukaryotic genomes are massive, they are actually most made of junk, right? So they're mostly made of defunct transposable elements, pseudogenes, different repeat elements, repetitive sequences, microsatellites, mini satellites. And here, for instance, here's the human genome. Only 1.5% of our genome encodes for protein coding genes. And again, we could talk hours about the different estimates of the number of protein coding genes in humans. They used to vary from anything between, I guess, 10,000 to up to 100,000. But in fact, currently we know that the human genome has around 19,000 to 20,000 protein coding genes. So most of our genome is actually junk. And later on, I want to talk more about this particular fact. Here, I'm showing you the number of protein coding genes in different eukaryotes, particularly in different vertebrates. So we have here, for instance, right? So humans, chimpanzees, mice and rats and cows and elephants, zebrafish. So different species here. Chicken. What we can see is that there is a relatively, diverse range of protein coding genes in those genomes, but that the number of protein coding genes does not explain organismal complexity, right? So I think everyone would agree that a zebrafish is less complex than humans. And look at this, zebrafish has relatively more genes than we do have. So it's clear, I think the community has come to terms with the fact that protein coding genes, the number of those genes does not explain organismal complexity. Now, diving a little deeper into the gene level aspect of things, I want to draw our attention to the difference in complexity also in terms of the gene architecture. So prokaryotes, as I've said before, their hallmark really is operons. We have genes sitting side by side, they're juxtaposed to each other, and oftentimes they're under the control of one common regulatory element, right? For instance, the Z operon. For eukaryotes, alternatively, we have for the lack of a better word, a real mass, a real genetic mass in terms of organization. So genes are broken down in axons. So they are actually, we have introns now. And in fact, for instance, speaking of the human genes, Our genes are long, but most of their length is because of introns, which you might remember. Those introns will need to be spliced out to give rise to the transcript, the mature transcript that will be translated. We have untranslated regions in those transcripts that serve regulatory purposes. But again, here I'm trying to represent to you the complexity at the gene level, how much regulatory, if you will, burden, how much baggage this has been accumulated. And later on, we can talk about evolutionary theories that try to explain these details. Now, going to a higher resolution. So we were here at the gene level. Let's take a look at the nucleus. We do have, so there is a whole field of genomics that studies genome topology and 3D configuration. There is actually a whole swath of techniques, very elegant techniques that study those things. And one I think the field has come up with, has really understood, is that the chromosomes themselves, they're actually organized in what's called chromosome territories, right? So within each nucleus, in different species, not only humans, okay, we have those chromosomes not in the metaphase, but in the interphase. They will be usually in their own territories. And you can really zoom in, just like we did with prokaryotes. You can take different resolutions, different levels, and analyze not only genes, as we did here, or not only chromosomal territories, but you can take a look at TADs, which are called, I will forget what they stand for, but those are domains, which are larger regions of the genome that are under common transcriptional control. Or they will also respond. We have heterochromatin, for instance, we have euchromatin. So you can take a look at those genomes in different levels. Now, and so if for prokaryotes, we had the tour de force of evolution in prokaryotic genomes was horizontal gene transfer, right? So horizontal gene transfer in prokaryotes really give them this versatility, this metabolic versatility and prowess, right, to adapt to different environments. In eukaryotes, the tour de force of genome evolution would be duplication, both segmental duplication, which is more at the gene level. So we're talking about gene duplication events, but also whole genome duplication events. Here I'm showing to you a few ancient whole genome duplication events. We have clear signatures of these events in the genomes of several eukaryotes. In fact, the first ancient whole genome duplication event to be discovered was with Baker's yeast. In comparison to other yeasts. And eventually, as more and more genomes were sequenced, we realized that, for instance, whole genome duplication events are relatively common among land plants, but we did have at least two major, and again, ancient, because they happened, millions of years ago, ancient whole genome duplication events at the very base of vertebrates. And then we have a third round in teleosts. So again, while horizontal gene transfer is the tour de force, is the hallmark of prokaryotic genome evolution, in eukaryotes, we're talking about duplication events. They can be either whole genome duplication or segmental local duplication events. Oh, I'm doing well with time. That's good. So now let's move to a little bit about genome evolution. And again, I hope that up until now, I've shown you, pretty much the field of genome evolution in different levels, right? And now I want to talk a little bit about, very briefly, because I'm certainly not an expert, but very briefly, a little bit about the pop gen of those genomes. So here I'm showing you what has become actually a very famous set of graphs not proposed, but published and demonstrated by Michael Lynch, who is a very famous genomicist. And what we can see here, so he has plotted genome sizes, right, on the x-axis against the ratio of certain genome elements. So we have, the ratio of coding DNA, the ratio of intron DNA, the ratio here of intergenic DNA. All of this is in log scale, right? And he has plotted this here. So we have prokaryotes, eukaryotes, we have here viruses. In eukaryotes are divided in different groups. What we can see, and here, these are called isoclines, so whatever falls within this or close to this, that means 100% of this genome is coding DNA, for instance, right? And what we can see is that so we have, as genomes grow in different groups, the number of coding DNA also grows, right? So they stay, again, relatively close to 100%, but then suddenly you have this curve plateaus, right? And we fall right here. So suddenly, as genomes grow, they're not accumulating more genes. They're actually accumulating more intergenic region. So you can take a look at the orange dots here, right? So take a look at those. So these genomes around here, 3 gigabases, only 1% of them have, codes for proteins just like ours. And then you fall here, most of them are intergenic. And a similar pattern here happens with introns. So intron DNA, which is, most of it is non-coding. It also accumulates as genome grows in different groups. So there's a lot to be said about this pattern, right? One of the many evolutionary hypotheses that tries to explain this pattern is the drift barrier hypothesis, which actually goes hand in hand with the mutational hazard hypothesis, and I'll talk about those now. So the drift barrier hypothesis states that we, genetic drift, which is represented with these arrows, is in a tug of war with selection, right? And as populations grow, but not census population size, we're talking about effective population size, which again, we can discuss those concepts in a second in the Q&A, but as the effective population size here grows, selection then becomes stronger and drift alternatively becomes weaker. So the evolution of the phenotypes of the traits can get closer to the theoretical maximum of that component. So here, it's called molecular perfection, meaning, so let's say within this area, selection is really strong. Selection can see any little, detrimental mutation, can purge that mutation away. So we are always close to what's the fittest version of that trait. Now, in smaller populations, selection is very weak because, or let me put this way, relative to drift, selection is weak. I think that's very important to remember. So in smaller populations, drift is strong, and then we can have the accumulation of what seems to be detrimental, right? So let me just go back to my, wait a minute, because I'm going almost in my final slide. I just want to go back here. So this complexity at the gene level, it is easy for us to realize that there is a burden in there, right? So the more complexity you add, both in terms, the more elements you add, easier it is for things to go wrong, right? Because for instance, for these introns to be spliced out, even though within the intron itself, these regions are evolving neutrally, right? They don't need to be selected for anything. There are a few motifs that need to be selected for to allow for these regions to be spliced out. So you are adding, it's almost like a ratchet-like system in which you add burdens. And this is what the mutational hazardous theory proposes, right? So in populations with smaller effective sizes where selection is weaker compared to drift, this is when mutationally hazardous elements like introns can be fixed in populations. And guess what? If we go back here, watch this. If we go back here, these groups of species that accumulate intergenic DNA in introns have small effective population sizes. So in terms of population genetics, this is when the environment that is suitable for the accumulation of elements of traits that are mutationally hazardous, right? So they can only be fixed when selection is weak compared to drift. So that's the idea of the drift barrier hypothesis, which goes hand in hand with the mutational hazardous hypothesis. Now, to wrap up, one thing I wanted to say, and I'm looking here at my other screen because I had the note here, I wanted to say this. Again, for this lecture, I didn't talk much about more population genetic aspects, such as mutation rates, recombination rates, and even drift itself. I think those topics are worthy of one lecture each, so we didn't have time here, but I want us to really have this in mind. When looking at genomes and genome evolution, we need to have this population genetics perspective to understand what is driving the patterns we can identify when looking at the genomes at the anatomy level. And to finish off, because this would be my last slide, I want to bring your attention to a very simple test, which is called the onion test, which is, I love that it's simple and elegant. And I think usually, in science, those are the most beautiful concepts when they are simple and elegant and really difficult to table, if you will. So the onion test was first proposed by Ryan Gregory, who is that Canadian genomicist I said. He's very famous in the field of genome evolution. And when there was a lot of, discussions on whether non-coding DNA is useful, whether, we haven't really identified the right signatures for function, whether each and every single nucleotide in the human genome has been selected for something special, Ryan Gregory came up with the onion test. And I have here a picture of the original web post, and I have here the link to the web post. I invite you to read the whole post. It's very well written. But here it goes, right? So the onion test is a simple reality check for anyone who thinks they have come up with a universal function for non-coding DNA. Right? So if you think non-coding DNA is important for structural purposes, or is important for regulatory purposes, or is important for coding for hidden proteins, you should run your hypothesis through the onion test. So whatever your proposed function is, ask yourself this question: can I explain why an onion needs about 5 times more non-coding DNA for this function than a human? Because the nuclear genome of onions is 5 times larger than the human. So if I'm saying that the human genome, for instance, all these intergenic regions have been selected for something special, well, guess what? What have they then been selected for in the onion, right? Why do onion and related species have much more, non-coding DNA than us? So I really think the onion test is beautiful, it's very elegant, and actually it applies to other types of genomes as well. So the organelle genomes, when you compare the onion mitochondrial genome with the human mitochondrial genome, you can ask the very same question. The onion mitochondrial genome, which I don't recall right now the size, but I know it's much larger than the human mitochondrial genome, which is our mitochondrial genome is just above 16,000 base pairs. You can ask why then, right? Why is the onion mitochondrial genome much larger if everything is being selected for, everything has an adaptive value, right? So the onion test is very, very eloquent, very powerful, and I would love my students to carry that concept with them throughout their careers. Well, with that, I want to say, so here I have a list of references. I will provide these slides to everybody so you can take a look and read more about the things. Each and every slide, you might have realized I have a link to what I'm talking about, right? So here is a abbreviated version of the references. Here are the, extensively written references, if you will. And just wrap up, if you want to study a little bit more about this, I highly recommend this book written by Lawrence Morin, What's in Your Genome, is a recent book. It's been written to the general audience. Very, very well written, very easy to follow, and very eloquent. And if you like to watch YouTube videos, I highly recommend Zach Hancock's YouTube channel. He has a series of lectures on evolution, both at the genome level, but also in different levels. So I highly recommend his YouTube channel. And I also recommend any book or paper written by Michael Lynch. Now, well, with that, I would like to thank you all for your attention. I'm in 40-minute mark, so I'm happy that I didn't go over time. Thank you, everybody.